Okay, thanks. Hi, good morning, everyone. Thanks for the introduction. And today I'm going to talk about how I use NIRS during my PhD to investigate the frontotemporal-parietal connectivity in awake infants. So I'm sure all of you are very familiar with resting state networks, so I don't want to go into much details of that. And among all the functional networks that have been studied, the network that I was particularly interested in has always been the 4-mode network, because this network has been related to mentalization skills and abstract self-processing, which are core topic of my research. Interestingly, the development of the 4-mode network is still something that is ongoing in research, and there's this interesting study by Trussens 2007 that show that while visual and primary sensory motor networks are in place from very early on, suggesting that these functions are already perfectly working from birth, development of the 4-mode network is actually gradually developing after the first years of life. And again, this is interesting because it suggests that the gradual development of the 4-mode network is actually related to the emergence of the social cognitive abilities over the first years of life. Most of our knowledge from functional network, the 4-mode network, comes from MRI studies which are normally done with asleep participants. But this has some limitation. The first one is the very high sensitivity to motion, and also the fact that usually, as I just said, participants are tested asleep. So that's why we think that fNIRS might be a quite good alternative to fMRI. In fact, with fNIRS we can test participants while they're awake, and this is quite important for us because as we heard yesterday in the wonderful keynote lecture in the afternoon, changing sleep stages can affect actual functional networks. So the fact that with NIRS we can test awake infants is quite important for us to have a quite precise estimation of the functional networks. And for those of you who are not familiar with NIRS, this is like our setup in the lab. So it's a quite small machine compared to fMRIs, compact, robust motion, and it's quite participant friendly. As you see here, the participants can just sit on the mom's lap. Just a very brief introduction of NIRS for those of you who are not really familiar with this method. So with NIRS we take advantage of, it's really similar with fMRI, to be fair. So we take advantage of the fact that oxygenated blood and deoxygenated blood have different properties, but in this case we use light to measure brain activation, and in particular we use light in the near-infrared spectrum. So oxygenated blood and deoxygenated blood have different colors. Oxygenated blood is brighter. So flashing light, we just put some optodes on the infant head and a flashing light through the scalp reaching the infant brain, we can infer how much light is absorbed, therefore estimating whether what the light reaches was oxygenated or deoxygenated blood, and therefore inferring brain activation. So one of the main weaknesses of NIRS is that we cannot reach with light very deep into the brain. So this is what we call the path of the light, that is usually called like banana shape. So this is like a source of detector. The light goes through several layers, skull, CSF, and reaches the detector. We cannot record very deep into the brain, which means that we have to study, I had to study only some portion of the 4-mode network as a proxy of it, in particularly focusing on, for example, medial prefrontal cortex and TPJ, which we know are two core regions of this network. Quite importantly, then I co-registered the optodes that I used to record this region on an MRI of the same age template to make sure to log exactly where we were measuring from. And here I'm going to quickly bring you through two studies in order to answer some important questions related to the development of the 4-mode network. So the first one is, could we measure the developmental trajectory of the frontotemporal-parietal connectivity as a proxy of the 4-mode network in awake infants? And the second one was, as we said, that the 4-mode network in adults has been linked to self-processing, whether we can redefine this link, the frontotemporal-parietal connectivity and self-processing in infants as well. So the method that we used was similar to studies. So as you all know, when you study, like when you acquire resting state with adults, you just say to your participants, line the scanner, think about nothing. And of course, we couldn't do that with our infants and toddlers. So again, this is like our setup. The infant or the toddler is sitting on the mom's lap. And we made up a screensaver-like video to keep the participants engaged, but not so particularly social stimulated. And interestingly, this methodology has been also recently used with adult studies as well, because it has been found that it kind of helps compliance and prevent to fall participants asleep. We then record like a long-lasting recording of this kind of resting state. So again, for those of you who are not familiar with that, every single line is a channel from where we recorded from. This is a quite very good participant, because as you see, all the lines are quite straight. We then clean the data from the channels that didn't have a good coupling between the skull, between the skull and the scalp, and then we based on behavioral coding, we removed every instance when the infant was not at rest. So either it was moving or someone was talking, anything like that. And then we performed the classical connectivity matrix between all the channels. And in order to answer the first question, so the developmental trajectory of the connectivity, we then acquire resting state at every 6 months, so starting at 11 months old up to 36 months old, and considering that we know that the brain is changing quite a lot over the first years of life, having this quite frequent acquisition, we thought could allow us to get quite precise estimation of the connectivity. And what we see here, I'm sorry that the image is not very high quality, but in green you can see connections that are increasing with age, and in pink you can see the connections that are decreasing with age. Here we plotted both oxygenated and then deoxygenated blood, which in resting state should have a quite similar pattern of connectivity. And we see that actually connection between every time points are increasing up to more or less the second year of life, where the number of connections that are increasing and decreasing between the 24-month-old and the following points are more or less the same. And again, there's quite similar results if we put all the subjects together in a linear mixed model. So if we estimate all the changes between the time points, we see that these are the connection within the fronto-temporoparietal channels, and these are reaching more or less their peak between 24 and 30 months old. So this is one of the first studies that estimated resting state in awake infants and toddlers, so we kind of provided evidence for fNIRS as a good tool to explore connectivity in awake infants, and we found this longitudinal frontoparietal connectivity increment over the first 2 years of life, and we know that we cannot claim that we actually estimated from the entire default mode network, but this is consistent with the fact that the default mode network is shaping over the development. And we found this maximum peak at about 24 months old, again, which is consistent with some previous studies done with fMRI that show that by 2 years of age, the default mode network is quite similar to the one observed in adults. But as you remember, at the beginning I said that I was quite interested to the fact that the default mode network has been linked to the abstract self-processing in adults. So my next question would be whether we can use this measure of connectivity to test this in infants as well. So as you can imagine, it's quite hard to estimate, to study the emergence of the sense of self or self-processing in infants. And one of the gold standard, one of the tests that has been currently used is the mirror self-recognition task. And here I have a video to show you how this is performed for those of you who are not familiar with that. Oops. There's no sound. But anyway, you can just read.
And we're really interested in looking at their reaction in front of the mirror. If they go for touching their face, it's a kind of index, a marker that they know how they look like, they know that there's something weird going on their face.
So as you see here, it's between usually 18 and 24 months old.
Play with them in front of the mirror and then pretending to wipe their nose. They put just a red dot with lipstick on their cheek.
Anyway, so it's usually between 18 and 24 months old that infants start to recognize themselves in the mirror. And performing this task, the experimenter is requested to classify the toddlers either in recognizer if they clearly touch the dot, ambiguous if they just stare at the mirror, or any other behavior falls in the non-recognizer their category. And the thing is that this test has actually been claimed to be a marker not only of self-recognition, but of a kind of broader sense of self. So there's still quite a lot of criticism around this test, in particular about the in-depth interpretation of the test. So there are some stream of research that are kind of against this in-depth interpretation, saying that actually recognizing yourself in the mirror, it's only like being able to match kinesthetic information with what you see, while other people, like other researchers that are more like towards this interpretation of the test, saying that actually recognizing yourself in the mirror, it's an index of more a broader development of the sense of self, so linked with empathy, personal pronoun use, and self-representation. So what we saw is that if we can actually measure the fronto-temporoparietal connectivity as an index again of the default mode network, we can see whether that is related to self-recognition and whether we can say something about the significance of self-recognition, right? So we performed the mirror self-recognition task as well, and we took the resting state recording that we did at 80-month-old to have a kind of quite balance between probably subjects that recognized themselves and did not recognize themselves in the mirror. And what we find here is that actually recognizer at the mirror self-recognition task shows stronger frontoparietal connectivity than non-recognizer, which are here plotted in green, mainly in the right hemisphere, which again is consistent with what has been found in adults as well, both in oxy and deoxygenated blood. So with the second study, we showed this. We showed that the fronto-temporoparietal connectivity might be a marker of self-recognition, so we kind of confirmed this link between the development, the full mode network and the emerging sense of self. And this is consistent with the several studies that had been done in adults that link the frontal and temporoparietal areas to the sense of self in adults. Is consistent with the only study that has been done in toddlers to test this, that relates the Y matter TPJ maturation with self-recognition in 15-month-old. And it's also consistent with an interesting study done with chimpanzee that found that chimpanzees that pass the mirror self-recognition task also have an increase in right hemisphere frontoparietal Y matter connectivity. So if it is true that we know that in adults the development, the full mode network is related to the psychological self-processing, if the fronto-temporoparietal connectivity is a good index of the default mode network, this link in toddler might suggest a broader development of the sense of self and not only basic self-recognition already at 80 months old. That's it. Thank you very much for your attention.